In this video, we will begin by discussing how our cells use DNA as instructions for creating proteins. First, DNA, or deoxyribonucleic acid, is a type of macromolecule that is found inside the cells of living organisms. DNA molecules contain all of the essential information required for the growth and function of all organisms. For eukaryotic cells, most DNA are found inside the nucleus, which is referred to as the command center of eukaryotic cells. Genes, on the other hand, are the basic functional hereditary units, and there are many genes found on a single DNA molecule. Each cell in the body contains around 30,000 genes. Each gene contains a genetic blueprint to make an mRNA, which often are then used to make proteins through translations. However, in some cases, the mRNA could be the final product of the gene, and these mRNA often serve some enzymatic functions. The main question is, how can the information in DNA and genes be expressed? And this is one of the main topics that we will be discussing today. Proteins are arguably the most important biological molecules. Proteins have many key functions, including as enzymes, signals or receptors, cell structure components, and regulators of gene expression and cell division. For example, hemoglobin is a protein in red blood cells that allows them to carry oxygen throughout our body. Other examples include actin and myosin, which are proteins that give our muscles the ability to contract, and insulin, which is a protein hormone that regulates our blood sugar levels. Proteins are made through two separate stages. The first step is transcription, which transfers the genetic information of a gene from DNA to an mRNA sequence through complementary base pairing. After that, RNA is used as a template to produce a polypeptide chain known as proteins, and this process is called translation. To start off, we will talk about messenger RNA, or mRNA, a type of RNA molecule that is used to carry information from the nucleus of the cell to the ribosomes. To start off, we will talk about messenger RNA, or mRNA, a type of RNA molecule that is used to carry information from the nucleus of the cell to the ribosomes. Compared to DNA, mRNA is a lot smaller, as it is only a copy of a small part of the DNA and is only single-stranded. There are two types of nucleotides, ribonucleotide and deoxyribonucleotide. The sugar and base between ribonucleotide and deoxyribonucleotide are different, as ribonucleotide is made of ribose sugar and deoxyribonucleotide is made of deoxyribose sugar. It is made by transcription, which is the process of reading our DNA. The process begins with an enzyme called RNA polymerase, which selects a single template strand of DNA to start the formation of mRNA. Nucleotides, which are the pieces that make up the code of both RNA and DNA, are connected into a chain by RNA polymerase. The template strand of the DNA uses complementary base pairing to make mRNA. The code contained within the mRNA is exactly complementary to the template strand. Next, we will discuss translation. Translation is the process of making protein at the ribosomes by reading the mRNA. The sequence of nucleotides in mRNA is made of triplets called codons. There are 64 possible codons, including one codon that tells the ribosome to start translation, and 3 codons that tell the ribosome to stop translation. Every other codon corresponds with one of 20 amino acids, which are the building blocks that make up proteins. Once the ribosome starts translation, transfer RNA or tRNA molecules bind to specific amino acids on one end and match their anticodon to the correct codons of mRNA on the other end through complementary base pairing. Based on the information coded in the mRNA in the form of codons, each amino acid gets added to a growing chain called a polypeptide. When translation is finished, the polypeptide will fold up into a completed protein. The environmental impacts on genetics are known as epigenetics. Epigenetics do not cause genetic changes, but instead changes the way the human body reads the genetic sequences. When humans are in their early years, their DNA contains a set of marks that are known as epigenome. The marks will change according to the experiences of the child, as it will change the way the genes will be carried out. Positive and or negative influences can have temporary or permanent signatures on the children's genes. This is why it is extremely important to make sure the children have positive experiences as a child, since epigenetic changes are most prominent during the early years of a human. Genomes are a living organism's complete group of DNA, so it contains all the instructions and details for the growth of the organism. DNA is the database of all the functions in our bodies, so genomics is the study and understanding of all the DNA sequences in an organism. Through genomics, scientists will understand exactly how genetics are caused and how to prevent the rise of genetic diseases in human society. It will also guide researchers and doctors to diagnose the diseases more accurately and provide patients and their family with a more humane, effective, and comfortable tactic to cope with those diseases. The Human Genome Project, which is considered as one of the greatest scientific projects in the last century, was launched in 1990 and completed 13 years later in 2003. Through the use of revolutionized DNA sequencing technology, the product produced the first complete sequence of the human genome, which later provided significantly valuable information for human biological, physiological, and genetic study, while greatly improving many medical procedures. Finally, we will dive into biotechnology. There are many different forms of biotechnology in the present day, but the one I will be diving into is genetic modification, specifically in the field of agriculture, and we often call the products genetically modified organisms, or GMOs. Genetic modification is a technology that transfers the genes of certain organisms to other species, transforming the genetic material of the organism. In order to improve the shape, production, and nutritional or consumption qualities of food, the technology of GMO can be incorporated into the production of food, and such food is called genetically modified food. The characteristics of genetically modified crops are similar to those of the original cultivars, but some of these crops are more effective against herbicides or pests, such as soybeans and corn, while others can slow down the softening and decaying processes of the tissues, such as tomato. Thus, GMO usually has a much higher production rate. American scientists first transplanted a gene that inhibits the softening enzymes in tomatoes into tomato cells and cultivated it into a storage-resistant transgenic tomato, whose growth period is more extended than that of ordinary tomatoes. As a result, the genetically modified tomatoes can be transported throughout the United States without the risks of rotting. In most cases, there are two ways to make transgenic plants: bacterial infection and technical means to shoot DNA into plant cells. First of all, the way of bacterial infection is to take advantage of some characteristics of bacteria and viruses, such as anti-insect or other characteristics, and the means of infection is to put the virus into plant cells and become a part of it. Another way is to coat the desired DNA around metal particles and bombard them towards the plant cells using a particle gun. Some of the metal particles coated with the desired DNA can possibly enter into the nucleus of the plant cells and become a part of the plant genome. Despite the higher productivity of GMOs, people have had a long-running debate on various problems caused by genetically modified foods. As a new biotechnology method, the immaturity and uncertainty of genetic modification will inevitably attract people's attention. Thank you so much for watching my presentation, and this is the end.